I have a great deal of pleasure being invited to this conference. And first, I would like to thank Mr. Zarbast and his fantastic organizer for the opportunity to visit this fascinating city. I would like to talk today about DNA diagnostic methods based on microarrays. I think it's a relatively advanced and probably interesting topic. I have to say, I am not a clinical researcher. I'm not doing DNA diagnosis whatsoever. I'm interested in the technology. I'm a biochemist, and we're looking at the identification of drug targets, for example. So we're looking at diagnostic methods on microarrays. Next slide, please. So as introduction, I would give you an overview on the topic of personalized medicine. For some years, we see now the advent of the so-called personalized medicine. This involves the integration of genomics, proteomics, metabolomics, molecular medicine, of course, biochemistry, and a lot of other disciplines. It is seen with great hope that this might be of very good benefit to the patient. So what I'd like to do is briefly compare conventional medicine as we know it and the personalized medicine. The drug development is based on empiric observations first. So you need disease mechanisms, drug targets, you need very extensive high-throughput screening, very time and cost-efficient drug develop. So a drug develop takes maybe 10 to 15 years until a drug can really go to the market. So the process has very high attrition rates. Also, the information on dosage is obtained with large or sometimes relatively small cohorts of patients. That means you get, in the end, a one-size-fits-all result. A big group of patients, that's the recommended dosage, that's the recommended drug. And we have to consider that the disease processes, in general, are epigenetic. So they work with proteins, and the therapy actually always aims at modulating aberrant protein function, but never the genetics. Then you see personalized medicine. The objective is to assess risk factors before the disease begins. You can use a lot of genetic analysis based on SNPs, polymorphisms, or even sequencing of genomes or large genomic elements. The advantage is that this allows you to predict individual risks to disease development. You also have so-called companion diagnostics. That means polymorphism, genetic data, gene expression are combined with protein function and clinical data. So the aim is a tailor-made preventive treatment. You can, by using this method, also look for susceptibility or tolerance to certain drugs. It is also possible to look at individual responses to dosage. And information is also obtained by non-invasive methods. And the whole thing integrates knowledge from many fields, as I said. Next, please. So in order to do this, you need, of course, a technology. And that's what I would like to focus on, microarrays. So that principle is always based on complementarity. With nucleic acids, it's obviously hybridization. And this is put into a microarray compact device. You have immobilized molecules, synthetic oligos, PCR products, proteins, assembled in an addressable format. You can then expose unknown test substances and examine where these molecules were captured. And you can have information on identity and amount of the molecule. Next, please. So this shows you the broad applications of microarrays. The first is the profiling of gene expression. You can obviously look into disease mechanisms, but you can also see tumor-related changes in gene expression. You can look for the effect of pathogens or response to treatments and drugs. The second I would like to talk about is SNP, single nucleotide polymorphisms, and so-called, generally called genotyping. You can thereby assess disease predisposition, complex disease traits, multi-gene effects like cancer, diabetes, Alzheimer. It also allows you to look in cancer at germline somatic mutations, and also very frequent in some types of cancers, loss of heterozygosity, LOH. Liver cancer, for example, lung cancer, often displays this is detectable by this methodology. Two other applications is just identify genes, like organisms in food, genetically modified organisms. I will not talk about this. And also sequencing. This is a topic of another talk. I'm not going to talk about it. What I would like to talk, if there's some time left, is protein interactions and protein microarrays. That is becoming increasingly important, especially because, as we know, the drug targets are proteins. And it is not, unless you consider some specific gene therapy, but the drug target is a protein. The advantage of all microarray technology is that it can process a very large number of samples. And that means you obtain massive parallel information on what you have in your sample. Next, please. So this is the first well-known example how this technically works. That is the gene chip from Affymetrix. That includes now all about 50,000 known human genes. It's a bit difficult to see here. As the first of this one was introduced to the market in '94 and was used for HIV genotyping. So the first genome, complete genome on that chip was Saccharomyces. But that was already in '97. So we are here now at about 400,000 spots on a very small area. We have two things what we need, basically. One is the so-called probe. The probe is what is on the chip. That is a very small amount, down to 1 picomole, for example, of DNA, which is bound to a metric surface. The metric surface usually used in this technology is glass, but there is another technology from Illumina, which uses microbeads, just to mention. You can then detect the interaction by fluorescence emission, and you see signals which depend on base pairing, temperature, annealing conditions, experimental conditions. we also have very broad range of coverage. The coverage or density on a chip is simply the number of representative probes. So that can be very small. Theoretically, it can be only one, because if you're interested only in one gene, then that's enough. So 1 to 3 for gene expression, but it can go up to 10,000 or 100,000 if you do large chromosomal assays, so-called tiling arrays. Next, please. So here you see an overview on expression arrays that uses immobilized oligonucleotides. These can be synthesized already on the quartz surface. So the relatively short, 25 MERS bioinformatics, 60 MERS, and you have then so-called spotted microarrays. That means your oligonucleotide probe is chemically synthesized and then spotted onto the surface. Many laboratories now prefer the so-called in-house printing. That means they make everything by themselves. The advantage is you just use what you want. You don't need expensive commercial chips, and it's very cost efficient. There are many labeling methods, actually. Biotin, streptavidin is very common. And now there's less expensive plastic-based technology available. So commercially, you have a wide range of products, Illumina, GE, Applied Biosystem, Eppendorf, so you can choose according to your experimental needs. Next, please. This shows you the outline, very brief outline of the experiments. Usually in expression arrays, you always combine or compare two conditions. This is like cancer cells, normal cells, disease-affected tissue, healthy tissue, but you can also compare samples from drug-treated, not treated, and so on. So then you just do the labeling of this. You have actually several choices depending on what you prefer. Either you make cDNA, you can do in vitro transcription, for example, or you can directly label any cDNA with fluorescent dyes. There's also a choice for the detection system. There are two basic detectors. One is the dual channel, and one is the single channel. The single channel, of course, is cheaper and is efficient if you look at relative limited number of samples. If you do large screening of samples, the dual is more efficient because for comparisons, you need twice as much arrays if you're using a single channel. Okay, next please. This briefly shows you, again, you start with extraction of RNA, labeling, determine expression level. You have two well-introduced fluorescent labels, Psi5 and Psi3 UTP, which are directly incorporated into the RNA, for example, difficult to see, RNA polymerase. That's important that you must compare samples of interest with a reference. These references are usually called spike-ins because the technique measures a difference in ratio. So you need to add a known concentration of reference substance to calibrate the result. Next, please. So this is a typical result from the literature. expression data. You compare lung tumor versus healthy tissue. The raw data show you the values for size 5, and for the labeled is size 3, the signal value. And then you see that a relatively large number of signals display up-regulated mRNAs for gene expression. So these raw data have to be treated with some caution. If you do expression analysis, you have two principal problems. One is that always non-specific mRNAs hybridize to the target. The second is if you use any amplification method, the amplification can be biased because some RNAs amplify better and compete out the less better amplified. Next, please. You know then obviously which probe is on your array. So you can then identify in your list, maybe let's say you use the entire human genome, then you get all these thousands and thousands of proteins. You see that, and the result is cyclin-dependent kinase, tumor expression-related proteins, numerous others are found to be upregulated. And that provides the first clue what might be an interesting target for drug development. These data require relatively intensive and laborious data processing by statistical software. And there is also a standard which is called Miami. It's not Miami in the US, it's with E at the end. That means the minimal information about a microarray experiment. Journals in which you want to publish microarray data require a checklist of procedures to eliminate a very wide range of errors. So you have to be aware that from the raw data to the real result is here a long way. I cannot talk about this in detail. Okay, next please. So this is then a real heavy result. What can happen if you do expression? This is targeted microarray for breast cancer. There's a total of 1,750 genes or so, which are simultaneously assayed on an array. And we cannot see all the colors here, and it is just to show the principle. You get this so-called heat or color map that shows usually the upregulated transcripts in red, the downregulated in green, transcripts related to the median level are black. And finally, you can construct a so-called dendrogram based on all these data, which simply shows the similarities or dissimilarities between your sample. So this is already very powerful example of a microarray aiming at expression, gene expression. Next, please. I will now go to sequence polymorphism, single nucleotide polymorphism. This is commonly defined as DNA variation at the base pair level. So it is found at the frequency of 1 SNP, so-called SNP, per 100 to 300 bases. And the whole set of genetic variations, or the sum of all SNPs in a population, is referred as the variome. This is now becoming increasingly important because we realize that the population have more and more polymorphisms, and that we see even individuals becoming very, very different and diverse on a genetic level. So no wonder that there's very great interest in analyzing this. We have so far, until last year, I think, 10 million SNPs described in humans. The SNPs are not just random mutations. There is a bias when SNPs are created in nature. More than 60% of all SNPs display a C to T replacement. And then only a few all other bases replaced. You see that 60,000 SNPs about fall within exons. The rest are in introns. Also the majority actually is in introns. But that doesn't mean that the SNP has no effect. Even if the intron is not expressed, but intron sequences contribute to splicing, for example. They contribute to regulatory sequences where protein bind. You have a lot of function for introns. For the SNPs in the exon, of course, they change the polypeptide sequence of a protein. And then we have only two types. We have peptide sequences unchanged, so-called silent mutations, but it's not a mutation, it's a SNP, called synonymous polymorphisms. And you have also different protein sequences, replacement polymorphism, and that's the great majority of all disease-related. Next, please. This shows you just the concept, because we have in the genome a very large number of SNPs, but it is not necessary to analyze them all. If you have what is shown here, 4 alleles of SNPs, you get haplotypes, and it is only necessary to screen the SNPs which clearly differentiate the alleles. Any other are pointless to analyze. So you can reduce the number of analyzed SNPs to a great extent. Next, please. So here you see the applications. Of course, this is now one of the key factors for the personalized medicine. The other key factor we are doing, we will hear later, is, of course, large genomic sequencing. Both go together, of course. And you see the result with SNPs are the so-called genome-wide association studies, the JWAS. That collects all informations on SNPs related to diseases. We have recently in our laboratory done a genome-wide association study for thalassemia, and the result was surprisingly, we found almost several hundred SNPs and could identify several hundred genes probably somehow involved, but we now need the genetics and biochemistry to look what these genes are now actually doing, because it's not known so far. You can assay, of course, response to pathogens, infection, chemicals, drugs, vaccines. I have to say many other methods are available for SNP analysis, depending on your experimental need. Yeah, you can do sequencing, mass spectrometry, capillary electrophoresis. They all have their pros and cons, and as I said, it depends on your experimental strategy, what you actually want to achieve. Next, please. So this is just the commercial system. This is the gene chip human SNP mapping assay produced by Affymetrix. Now, it's certainly, we have 100,000 SNPs per experiment already, and the methodology is commercially available. I've never used it. I can't say what the price is actually. I guess it's relatively expensive. Next, please. Then you see several scientific information resources which you can use to analyze the result of all these experiments. One is the HapMap project. That stands for the haplotype mapping of all genetic variations in human, actually. The haplotype